here.
Glucose.
So the glucose, maybe not the pure glucose, but it's going to be some sugars. It could be fructose or it could be sucrose, which is disaccharide. Fructose is a monosaccharide. Like I said, glucose is mostly it's a grape sugar. So, you know, if tomatoes are sweet, well, in the onion is going to be sugars also, by the way. So definitely it's going to be simple sugars, monosaccharides and disaccharides. Sucrose and fructose there. What else?
Fiber.
Fiber. Fiber belongs to which group? To what group fiber belongs to?
Is that cellulose?
It's a cellulose, yes. Belongs to what big group? We started with this group classification. Can't hear. Maybe you're right, I can't hear.
Carbohydrates.
Carbohydrates. And cellulose is small molecule or big molecule? Big molecule. So that is a polymer made of the same monomers that starch and glycogen. They are same monomers. We're just talking about monomers of starch, monomers of glycogen. Glucose. So the same, but it's not available for us. We cannot choke cellulose fiber and get that glucose as our blood sugar. It's not digestible. For cows and rabbits, yes, but not for us. But still we need that fiber. Okay? Simple. You consume them. Every day we covered carbs, we covered lipids, and we covered proteins. Okay, so now I can see a couple students who missed, and you need your papers here. So that is outline for test, outline for lecture, and then it's going to be description of your this assignment. And I have your labs, and just don't leave after the class, okay?
Thank you.
So that's 3 papers. Outline lecture, this description, we're going to talk about it now. And no, this one. Okay. Okay, we've done it now. So the quiz is going to be about organic chemistry. So now about your assignment number 1, I will try to finish everything that is included in your test number 1 during this week. And then during the weekend, you will have the possibility to do your cell model. And the next week, well, if I couldn't finish everything, we are going to take part of Tuesday to finish lecture, but then it's going to be your presentations. And this Thursday is going to be test. So, the first thing you have to decide, like I told you, which cell is going to be prokaryotic or eukaryotic. And if it's a eukaryotic, you are going to decide which one is going to be animal cell or plant cell, because they're different. I brought you some examples that I saved, not very big, not very heavy. Those are the easier to carry. The most unusual cell model was just so easy to carry. I had a Japanese student. They have that art of folding paper, origami. He made the model of plant cell. So that is like a cell wall. And then all these beautiful and small structures that are all organelles. Of course, I'm not going to ask you to do that, but I said that some people are very, very creative. So this is why some students like to do a prokaryotic cell, so which is on the screen right now. Because they think that is so cute to put all this pile around it. to play with that, you know, to touch it, to show that it is my prokaryotic cell. So it has plasma membrane, cytoplasm. Inside you can see ribosomes, they are not real organelles because they don't have double membrane. You can see that loop DNA, they don't have nucleus, so just DNA is hanging out in the cytoplasm, places called nucleoid region. So most of all these prokaryotes are bacteria and archaea. Some of them have additional cell wall, and some of them have even a capsule. They have even these appendages. The singular is going to be pilus. Many of them pili. They are for attachment. So and they have flagella many or flagellum. That is much longer appendage for locomotion, for swimming. That's it. That is what the student is going to come with that presentation. That is going to be what is going to be said. There's probably one question is going to be asked. So therefore, the prokaryotic cells are usually very small, 1 to 10 micrometers in the length, so the diameter depends on their shape. So they're very simple, primitive. They don't have organelles because organelles have double membrane. Ribosomes are not considered. Again and again, I'm talking about that because students always make this mistake. So ribosomes are protein synthesis. Of course, bacterium needs proteins. So, but they don't have double membrane, so they are not considered to be real organelles. So no real organelles and no nucleus. No nucleus, no organelles, small. That's it. 3 differences. So at the same time, I'm going through the stuff that I already covered. So then, well, I believe that actually it also looks like it is prokaryotic for me. So that, you know, again, so there's some students like to do knitting. So again, it's a flagella. And as usually the most common, it's when the just a styrofoam. But again, when it's going to be a plant cell, they don't need to even buy styrofoam. So that is for animal cell, definitely. Because it doesn't have cell wall. If it's going to be cell wall, use a shoebox and just put all these organelles there attached. What I'm going to tell you, my experience showed that sometimes students wanted to make something unusual, and every time it was a failure when they tried to make a jello in a big dish with all these organelles there. And when it doesn't set, so it's disaster. You cannot breathe it, you know, or you can just splash it all over your vehicle. So please don't do that because it usually doesn't work. Another creative case, it usually works. Sometimes students just bake, like a pizza or a cake. So, and they bring it here. So it's up to you. But I said, it should be something really, really simple. So therefore, this one, this example of animal cells, so you can say eukaryotic cell has plasma membrane, nucleus here in all organelles, like in the plasma reticulum, we're going to talk about that. This is rough endoplasmic reticulum, it's covered with ribosomes. This is smooth, not covered. Mitochondria, I can see lysosomes. And actually, there's even just, you know, for convenience, probably for presentation, even words here that just, you know, stickers attached. So again, it's up to you. So that is what is going to be your cell model, and plus you have to submit preferably typed list of organelles with a brief description. It's going to be only on 1 page. So therefore, that, as you can see, eukaryotic cells much more complex, much bigger. They are different from each other because all plant cells have, besides that they all cells have plasma membrane, and the plant cells have additional cell wall made of cellulose. By the way, when we're talking about bacteria, their cell wall is not made of cellulose, it's beta-glycan, and we're going to talk about that in microbiology topic. So, but if you're going to say cell wall, just don't say that it's cell wall because it's not true for bacteria. So, and for Okay, for animal cells, for plant cells, you have to put that nucleus, which is the command center of the cells, and main organelles like endoplasmic reticulum, cytoskeleton, mitochondria, of course, is going to be a chloroplast for plant cell because it's very responsible for photosynthesis. Also, it's not true that all plant cells do have chloroplast. Think about roots. Of course, it's not going to be chloroplast because the light is not available. Why bother? So, but those that are exposed to light, of course, and we like to show that plant cell has chloroplast. They're responsible for photosynthesis, and they have vacuoles because most of animal-like cells don't have vacuoles. So, if you have any questions, just don't hesitate and ask, but now we are going to go through these organelles. Then on your outline, you also have some part of from chapter number 5, passage of chemicals through plasma membrane, because it was time when I had to lecture about them sooner than about energy, because of lab that was earlier. But not this time, so I'm going to follow exactly like chapter number 4, cells and organelles. So we were talking about the size of the cells, and I told you that some cells are unusually big, that is eggs, birds' eggs, and those eggs that you can see, fish eggs definitely. You have it here, so, and the eggs of these snails, aquatic snails, you still can see them, and of course you can see birds' eggs. So they are unusually big cells. The rest of cells are extremely small, and most of them were discovered after discovery of microscope. We covered that. Who did that? And only in 20th century it was possible to find out about most of organelles, because when you're using your light compound microscope that you use during your lab, you're not going to see all these organelles. Mostly you're going to see, you're going to see a nucleus, and you're going to see chloroplast, pretty good. You're going to see vacuole. That's it. You're not going to see mitochondria, the plasmic reticulum, ribosomes. So they were discovered when electron microscope was finally created. Because everything about microscope is about resolving power. And resolving power, it's about distance that allow you to show two objects as separate dots. Like, for example, so our eye can see 1/10 of millimeter. So remember millimeter that on your ruler, the tiny distance between two lines? 1/10 is going to be a tiny dot. You can still see it. So the light microscope is going to have 0.2 micrometers, and the electron microscope is going to give us 0.2 nanometers. So remember that if we divide 1 millimeter into 1,000 quarters, it's going to be micrometer. If we divide micrometer into 1,000 subunits, it's going to be nanometer. So therefore, definitely, when electron microscope was created, it helped a lot the scientists to learn about organelles. So nucleus, which you're going to see because what is inside under the microscope, because it has a different optical properties. Even without special dye that usually makes it darker and more visible, we usually put some methylene blue, for example, or iodine to see nucleus better under the microscope. Inside the nucleus, it's a special liquid or gel mass that is called cryoplasm. So what is inside the cell is a cytoplasm. What inside the nucleus is going to be cryoplasm, because another name of nucleus is karyon. That is why we're talking about cells. Prokaryotic before nucleus, and eukaryotic means true nucleus. So inside the nucleus, so we have this cryoplasm, and then it should be another round body. Sometimes one, sometimes more than one. Depends on what kind of species we're talking about. These bodies, it's like a tiny nucleus, so therefore in Latin language they use a suffix. It's nucleolus. Nucleus, bigger, nucleolus, smaller. It's sitting there inside the nucleus, and it's responsible for making ribosomes. Those not organelles that all cells have in their cytoplasm, and they are responsible for protein synthesis. Ribosomes are made of proteins and RNA molecules. So therefore, nucleolus is very important part of the nucleus. It's very interesting when we are going to talk about the cell division. That is going to be mostly about chromosomes that are sitting in the nucleus. But before division, they are not visible because they are not condensed. They're so thin that we cannot see them. When cell is ready for division, they become condensed. They look like fingers or like weenies, and you're going to see them during your lab because they're going to be pretty much visible there under your microscope. But in a usual cell, you take it, you look at the nucleus, you cannot see any chromosomes, but they are there. And they're made of protein and DNA, and portions of DNA, portions of chromosomes, and genes responsible for traits. So definitely DNA dictates RNA synthesis. RNA participates in making ribosomes with the help of nucleolus. In a mysterious way, during the cell division, nucleolus disappears. When division is over, it's coming back. So again, what you can see, what is around the nucleus, it's called a nuclear envelope. That purple color, nuclear envelope. That nuclear envelope has openings that are called nuclear pores. They're like doors or windows, whatever you prefer to think about. Because DNA is a big fat boss that is sitting in the nucleus. It's an office for that DNA. DNA is not going to go into cytoplasm to dictate how to make proteins. But RNA as a secretary is going to take information from DNA and then go through these openings, nuclear pores. So whatever you prefer to think about, that secretary is going to go like a normal person through the door, or it's going to be some kind of special secretary that's going to jump through the window, going into the cytoplasm. But this is nuclear pores, just for RNA to go out of the nucleus into the cytoplasm to tell how to make proteins. Definitely what is going to be close to the nucleus, so I'm going to show you that is close to the nucleus. You can see something that looks like labyrinth, like some kind of folded canals. So they are called, these very important organelles are called endoplasmic reticulum. Very often, when you're doing your presentation, if you don't want to break your tongue, you can say ER, endoplasmic reticulum. So, and there are two types of endoplasmic reticula, which is plural, rough and smooth. A rough, it's rough because it's covered with ribosomes, and ribosomes are for protein synthesis. So the protein synthesis takes place on the rough ER, endoplasmic reticulum. It's like if you are trying to touch it with your fingers, and ribosomes like poppy seeds or like tiny beads, and you touch and it's really rough. So just make these images. What is difficult about that? You have to use your imagination. You cannot see it. That is why I'm asking you to do that model, and their purpose is for you to learn about this stuff. When you're doing this model, you put in stuff together, talk to yourself. Okay, now I'm putting in the plasma reticulum, so when I'm going to cover it with ribosomes, it's going to be rough. And what is it for? As ribosomes have protein synthesis, that is for protein synthesis. And also it looks like a big canal, so these proteins can be delivered from one part of cell into another. Then smooth endoplasmic reticulum, no ribosomes, also for synthesis, but not proteins. Molecular lipids, very important chemicals are going to be made there. So therefore, two types of endoplasmic reticulum. The biggest mistake students make when the question is in what organelle protein synthesis takes place, and that is one of the options ribosomes, and they're so happily, ribosomes are not real organelles. Rough endoplasmic reticulum is going to be the right answer. So this is subtle details are very important when you're taking tests. So where all these chemicals are going after they are created on endoplasmic reticulum, they go to the next organelle that is called the complex Golgi. That organelle is named after Italian biologist Camillo Golgi, who discovered this organelle and described it. It looks like also not completely like the plasmic reticulum labyrinth. It looks like probably stuff of envelopes of pita bread. And you can see some kind of spherical things getting out of that. So, so what is this organelle for? So chemicals after they were created on the plasmic reticulum, they go there. So it could be like a post office, or it could be an idea of some kind of warehouse where they have to sort out these chemicals and make packaging and even activation. So they're going to be activated and they're going to be packaging these vesicles, these bubble-like structures. They're going to be detached from that organelle, already prepackaged material. What can be there? For example, insulin hormone that has to get out of the cell. So it's going to be packaged in these vesicles. So they are made, because it's protein, they're made on endoplasmic reticulum, on the rough endoplasmic reticulum, but it's going to be activated, sorted out, and packaged into vesicles here in this complex Golgi apparatus organelle. So then, next organelle that we need to know, it's also like a spherical body. It's called a lysosome. Could be a lot of them in certain cells. Lysosomes have also double membrane. All these organelles that I mentioned, they have double membrane. Of course, even the nucleus has double membrane. Endoplasmic reticulum, double membrane. Complex Golgi, double membrane. And lysosome, double membrane. Inside the lysosome is going to be a lot of catalysts, a lot of enzymes. And this is a recycling center of the cell. When some organelles are getting old, that they cannot function, instead of just like throwing them away, they're going to this lysosome and it's going to be disassembled. And all these chemicals and parts of these organelles are going to be used then to create the new ones. The lysosomes is very important organelle for such interesting event that we're going to mention when we talk about animals, both invertebrates and vertebrates, as metamorphosis. when a tadpole is getting ready to become a decent frog, is going to lose its tail and gills. Instead of gills are going to be lungs. Yes, it's very complex event. So therefore, that tail is going to be taken out by lysosomes, and that materials from tail are going to be used by lysosome for, it can be used for something more creative in their future. So think about the pupa, and caterpillar is going to be converted into a beautiful butterfly. So definitely, it seems like it's not even living thing. That pupa is laying there, looks like an absolutely dead material. But a lot of interesting events are going there, metamorphosis. And believe me, those lysosomes are going to play a very important role there. The reason why I put these two very important organelles together here on the screen, mitochondrion and the chloroplast. Mitochondrion, all eukaryotic cells have them. Animal cells and plant cells do have mitochondria, and it's a power station. When we are in aerobic cellular respiration, biochemistry, we are going to talk about mitochondria. The most important events will take place there in this organelle. So chloroplast is for captured light energy. So green pigment chlorophyll is located there, and that light energy is going to be converted into the chemical energy. And sugar is going to be made from inorganic chemicals, carbon dioxide and water, photosynthesis. So a very important organelle. So both of them have double membrane. Both of them have inner content that is called the stroma for chloroplast and matrix for mitochondria. And what is very interesting, both of them have the ribosomes hanging out there inside that inner content, and both of them have own DNA. So scientists have hypothesized that these two organelles were prokaryotes, much smaller than eukaryotes, but then they got inside the bigger eukaryotes and stayed there, being converted during the evolution into organelles. So, own ribosomes, own DNA. Coming back here, you can see prokaryote. Yes, inner content, ribosomes, and DNA. So they say we have mitochondria and chloroplast. So we have ribosomes and we have DNA. Well, mitochondria has two membranes. Outer membrane is smooth, and that organelle will look like a bean or loaf of bread, whatever you prefer to think. Inner membrane is folded. You can see it's a lot of folding. Therefore, it creates a huge space between membranes. That is extremely important because we will learn about and make analogy to the hydroelectric power station when water is stored in a dam that is going down. That kinetic motion of water rotates the turbine, is attached to the generator, and we have conversion of kinetic motion of water into the electrical current. That is power station 2, but we are going to deal with motion of hydrogen ions, which are going to be concentrated like water in a dam between membranes. That is why mitochondria is called power station. But again, all these details are going to be, we're going to talk about that when we are in biochemistry. So now what you have to remember, double membrane, matrix inside, own ribosomes, own DNA, and idea, hypothesis of so-called endosymbiosis. And the inside seem together by life. So just smaller prokaryote got in a bigger eukaryote and stayed there being converted into organelle. So the same way chloroplast has own own ribosomes and own DNA, and we leave it for some kind of very primitive prokaryote with ability for photosynthesis. Yes, some bacteria are able for photosynthesis, the bacteria prokaryotes, and then they got inside into bigger eukaryotic cells and stay there becoming organelle. So it has these discs, green discs, they're called thylakoid discs, and on the membrane of them is located that green pigment chlorophyll, chlorophyll that is capturing light to convert it into chemical energy. A vacuole, that is going to be like a pond inside the cell, or lake inside the cell, or swimming pool inside the cell. Some kind of water body that is for storage of water and many other chemicals in plant cells, and for some other purposes in the protozoans. So therefore, all plant cells have vacuoles. Some plants have smaller vacuoles, some plants have huge vacuoles. You think about the juicy watermelon is going to have a lot of vacuoles, and what is going to be inside? Watermelon juice. So what is going to be there? What kind of chemicals are going to be in watermelon juice?
Sugars.
So it's going to be water, sugars, maybe a little bit of acids, and some pigments because it's pink, yes? So the same in the strawberry, the same in the strawberry vacuoles is going to be also sugars, acids, pigments, water. What about a lemon vacuoles? So it's going to be water, acids, some pigments, yes, a little bit yellows. What about in poison oak vacuoles? That oil that is causing allergenic reaction, that causing that irritation, burning sensation. So depends what kind of plant we're talking about. Well, and then you can see amoeba, which is protozoan, also eukaryote, also has organelles. You can see their nucleus there. And it has so-called contractile vacuole, because that is freshwater dweller, and very soon we're going to talk about osmosis. I will mention again that contractile vacuole then. So they gain water by means of osmosis. They don't want to be ruptured, to be overfilled with water. So they have this contractile vacuole that is going to remove excess of water. So therefore they're just different vacuoles for different purposes. So cytoskeleton. All these nice organelles inside the cell, inside the cytoplasm, they have to be organized in some way. And their cytoskeleton, it's a system of ropes, of fibers, thread-like structures. They are going to keep organized at a certain place all these organelles. And some of them have specific functions. So we can see some of them responsible for cell movement, because like you just saw amoeba, that amoeba is going to move because of flow of cytoplasm, and so it's going to be some fibers that is going to help with that motion. And then you can see on the bottom microfilaments, actin and myosin. So they are proteins in that filaments of muscles, so they're able to stretch and shorten, so that is responsible for contraction of muscle. Now then they have this microtubulus. They are going to participate in the cell division because they are going to make that spindle apparatus that are going to keep these first double chromosomes together, and then they're going to separate them because they are going to pull them apart, these two sister chromatids, and they're going to become two new chromosomes. So, and some of these filaments also make skin stronger and able to stretch when we grow, or for example, when a woman is pregnant, so the stretching of skin. And then some of these filaments make certain structures really strong, like think about wings of butterflies, or think about feathers of birds. So it's going to be a lot of different fibers there. So this is not going to be part of your test, so it's just like additional information because it's in your textbook, you're supposed to know about that. But mostly what you need to know about nucleus, about endoplasmic reticulum, chloroplast, mitochondria, complex Golgi, lysosomes, and vacuoles. That is going to be the most important, so for you to know. So now, again, like we were talking about this flagellum that prokaryotes have, eukaryotes, so we're talking about eukaryotes now. They also can have these appendages that serve for different purposes. So these big pictures with these bluish colors, you can see some flagellated organisms with a flagellum. They can move because of that whip-like organ that's going to move in a liquid. So some of them are parasites, and they are going to use this flagellum to swim in human blood. So like, for example, trypanosoma bursae. Just what is exactly below with that like looks like a couple of pink eyes, and but they're not eyes definitely, and some kind of purple color. So that is a Giardia I believe. So Giardia is parasite, and unfortunately is flagellated protozoan, parasitic. That unfortunately very, very common in our area. Our pets can get it, especially after rain. If they drink that water outside, and if during the night some wild animals were going around and they were pooping in different places, and that is parasites with their feces can get in the water. So the same way when you go camping and you forgot to bring your drink with you and you're so thirsty, and you can see that nice good-looking water coming in the stream, and you cannot resist, and you're coming home and you have your stomach ache and cramps and diarrhea, you got these parasites, you need to use antibiotics. So it's going to be pretty fast to get rid of that. The same about pets, but actually it's very unpleasant to have them. So they use flagella for locomotion also. Then to the left you can see the sperm cell, of course flagellum for the movement that sperm cell is going to use. So that is a whip-like appendage. Much shorter is going to be on this side. On this black square you can see some kind of a bloom creature that is covered with tiny like eyelashes structures that are cilia, and that is ciliated protozoa. So this one, parasite with flagellum, is flagellated protozoa. They even have their names because of their appendages. They have a flagella, and this one has cilia. Cilia is like a tiny hair-like structures, and they allow them to move. They are free-living, not parasitic. Live in fresh water, they can move, and they can also sweep food particles inside their single cell. So they're very important. We do have ciliated cells. This on the bottom, the pink, where you can see some lavender colored nuclei, and you can see that like again, so like a pink brush on the top. That is our upper respiratory tract, ciliated cells, we do have it, to trap dust. When you clean your throat, then you swallow after that. Actually, they collect that in their mucus, they collect dust that you get from air, and then you swallow it. And this all bacteria, all this dust goes into highly acidic environment of the stomach, and these bacteria are completely doomed. Instead of going in your lungs, they go in there. So that definitely, that is for protection. And another one on the top, that is about our ability to hear. So you can see outer hair cells. That is in our organ of cortex. So that is going to be, so you can see inner hair cell, that is going to be outer hair cells. And then in our inner ear, we are going to have similar cells. And you can see that is going to be membrane on the top. And hair cells are going to touch this membrane. And our ability to hear is due to the elasticity of membrane, which is diminishing with age, and with the number of these cilia of the cells, these hair-like structures, exposure to very loud sounds can just destroy the cilia. And therefore, when people play music in band and they don't protect them, or they use, for example, they go to practice shooting, and again they don't protect their ears, so they can definitely destroy their cilia, and their hearing is going down. And of course, power equipment, which is my husband's business, and he is a wonderful example. He didn't, when he was working with, all the streamers and chainsaws and tractors and all this. So, and when he didn't protect his ears, well, now he's a big boss, he doesn't need to do that, but he already ruined his hearing definitely. Of course, it's very selective. If you can say with a whisper, ice cream, you still can hear that. But you can see that he has to do something, immediately he cannot hear you. And you know the trick. Probably you know some of your relatives with a similar problems in hearing. Okay, so we're done with that. Since we still have time, I'm going to give you your outlines for chapter number 5, which is dedicated to energy. Please start giving it, and also start giving to everybody. And we are going to start talking about energy. And then this way we are going to finish everything next time, because we are going to talk about, we are going to use this outline, so don't forget it next time. We are going to use it because we are going to talk about the passage of chemicals through plasma membrane. And so probably a couple things I didn't have a chance to cover from that lecture about cells, just to make sure that I will do that. I will talk about the structure of plasma membrane when we talk about passage of chemicals through plasma membrane, so we'll think about that. The surface to volume ratio, that is lower third of your outline here. So surface to volume ratio. Well, since I put it there, I need to explain. So usually scientists say why cells are so small, because it's much easier for them to have communication inside the small cell. Because when we are thinking about this cell of this size and cell that is going to be 10 times bigger in the diameter, when you think about some kind of a hypothetical spherical cell, of course not all of them spherical, but we're thinking about diameter, so the surface of this cell is going to be 100 times smaller than this. So So diameter 10 times bigger, surface is going to be 100 times bigger, volume is going to be 1,000 times bigger. So therefore, that is explanation how to keep the surface to volume ratio by cells as small. And shape of cells is defined by their function. So that, for example, when we are going to deal with our epithelial cells here in the lab, not tomorrow, but a week from now. They are for covering and protection, so they have very good connection. They make that nice film inside our mouth cavity. It's actually, they cover all cavities in our body inside, like thoracic cavity, abdominal cavity. And that they are for covering protection. Fat cells, adipose cells, they look like big sacks or big, fat containers with milk, something for that has a real volume. They're for storage of fat. Like, you buy the container with, for example, butter or cottage cheese. So that is going to be analogous. So, and the muscle cells, they're really long because they're like a rubber band, because they're able to stretch and contract. Yes, please.
What outline are you talking about?
I'm talking about this, but I gave you about energy, but I said I'm covering what I haven't covered here. Yes, and still we have some things to do. We are going to talk about that matching of organelles, and we are going to talk about the diffusion osmosis next time. But the chapter number 5 starts with this, then it goes to the passage of chemicals. So therefore, this outline with the cells has materials from chapter number 4 and number 5. But now about energy, so the capacity to do work. And we are dealing with different forms of energy, and they all can be classified as potential energy, which is about non-movable objects, very often elevated, but depends what kind of. We are going to get some examples. And kinetic energy, that is about moving objects. So we're going to talk about light energy, mechanical energy, sound, heat, electrical current, and radiation. It's all different forms of energy. So therefore, this outline that I gave you about energy, that if you take your chapter number 5 and start making notes, so it's going to be approximately what you need. So it's really essential information is here on this page, because it's actually almost irrefutable. So therefore, potential energy, elevated objects have potential energy. So they're not moving, they are somewhere on the top. So, and you can see they say that those that, since energy is capacity to do work, and work is mass times distance. So mass is very important. Therefore, they say that which person has bigger potential energy, the bigger, the heavier. You don't need to memorize it. Just try to get some concepts about energy. You have potential energy. You are elevated above the floor. If you're going to fall asleep during my lecture, you're going to drop on the floor. That is going to be your work, your mass, and times distance to the floor. So you are going to hit the floor, but of course it's going to be your potential energy is going to be converted into kinetic energy of your body dropping down on the floor. Chemicals of food have potential energy. You can see that sugars in an apple there, they have potential energy. Because you can consume it, you can digest it, and they are going to be converted into ATP in your body, and there's going to be storage of energy. Chemicals of fuel have potential energy, so definitely it's very important. Then more energy of position, so like here you can see about that a spring. So it's going to be different forms of energy here. So potential energy, we're still talking about potential energy. And then kinetic energy, energy of motion. So somebody is moving, there was a elevated, have potential energy, now going down, so it's conversion of potential energy into kinetic. Light, it's a movement of particles of light, photons, of course it's energy, it's kinetic energy. Electrical current, of course, it's a motion of electrons, it's kinetic energy. And the heat, of course, it's about random motion of atoms or molecules, whatever, and moving from place to place. The hotter, the faster they move, of course they move, it's kinetic energy, you understand? Plus, heat always goes from what is warmer to what is colder. Of course, it's kinetic energy. Sound, again, the movement of air, yes, so different types of sound. So it's going to be examples of kinetic energy. So therefore, look around, give some examples of energy you can observe in our class.
You walking forward?
Yes, so you're breathing there, yes, so yes.
Light the most.
Light, definitely. We have electrical energy, yes. We have heat going from your bodies. We have a sound, definitely. So we have a lot of different forms of energy. Okay, so some examples of energy conversion. So that is very, very convenient, yes? So we can convert electrical energy into light energy. Electrical energy is pretty expensive energy. It takes about 300 units to give 100 units, but it's very convenient. It's so convenient because we can convert it in anything. We can start vacuuming, we can cook food, we can heat our place, we can ground coffee, you can just make your computer work, and so on. So that is why we are doing that very expensive. We are using very expensive form of energy, electrical energy. So where is our electrical energy coming from in our area? We have hydroelectric station. So therefore, again, one more time, I already covered it when I was talking about mitochondria. So we have potential energy of water stored in a dam, yes? Then water is going down, so that is potential energy of water stored in the dam is converted into kinetic energy of falling water. Then it's going to rotate the turbine, which is connected to generator. When generator, it's all kinetic energies, all conversions of energy. When generator is rotating in a magnetic field, it gives us electrical current. It comes here and we can convert it in all other types of energy. Two laws of thermodynamics. First one, and you have to know them. That's pure physics, but you have to know. First, energy is the law of conservation of energy. You have it on your outline. Energy cannot be created nor destroyed. One form of energy can be converted into another form of energy. So the same amount of energy in the universe. Like we're talking about conservation of matter, the same amount of matter in the universe. So the same amount of energy in the universe. People use these, when they talk about energy, they forget about it. They make mistakes all the time. You can hear it on the radio, on the TV, everywhere. They say, to make energy, to create energy, actually that is wrong. But they just prefer in daily life to use it. From the point of view of physics, you cannot say energy was created or destroyed. No, it was converted from one form into another. And what is interesting, that second law of thermodynamics says when one form of energy is converted into another form of energy, some energy is getting lost as heat. It's not possible to convert 100% of one energy into 100% of another form of energy. Always is going to be lost as heat. And since we always can talk about conversion of any form of energy into heat, and heat is energy, so therefore we can use some units of heat. So there's only 1 calorie amount of heat required to raise temperature of 1 gram of water 1 degree Celsius. For food, we use a calorie, but with the capital C, and it's equal 1,000 these small calories, just for you to know when you're looking at the calories. We always can talk from this point of view about high quality energy, and there's going to be atomic energy, chemical energy, mechanical energy. So atomic especially, a very small amount of fuel can do a lot of work. Chemical, yes, you can eat banana and it's going to be chemical energy, but starch and sugars is going to be digested and it's going to give you fuel, glucose, and you can survive for a while. So your fuel in your vehicle is going to move you from one place to another, so it's a high quality energy. It's going to be also chemical energy, so therefore definitely it's going to be mechanical. Low quality energy is going to be heat, because normally heat goes from what is warmer to what is colder. You cannot collect it and put it back and make it work. So therefore heat, and it's actually a result of every conversion of energy, as some energy loss is heat, so it's a low quality energy. So we're talking about heat is less organized form of energy, because it's going from what is warmer is going to what is colder, and you cannot get it and make it work. So a little bit more. Therefore, we're coming to the idea of entropy is a measure of a system disorder. We just said that heat is less organized form of energy. You understand why? So entropy increases, and you have to say without energy input, which is literally what you heard from your childhood. You cannot get anything without doing work. Nothing happens without work. Nothing is going to be built by itself without work. Nothing can become clean without work. Nothing can become organized without work. And work is energy input. You understand that? So from this point of view, you have to understand that you can see what is just probability that you toss in bricks and they're going to be organized. More likely they're going to be disorganized. You need to put them. You have to use some of your energy, and it's going to be some work to make them organized. So the same way about that heat going in all possible directions, becoming disorganized. And since every conversion of energy, and we're dealing with that all the time, it is going to result in some heat. It means everything is going to become more and more disorganized without energy input. But we have our universal source of energy, sun, that was with Earth for a long time, and they say that is going to be with at least 6 billion years, so not a source of energy. So therefore, definitely you understand without cleaning that, nothing is going to become organized. And if we're coming to the fall time, when there's going to be a lot of leaves going down, and sometimes you need to clean, you have to collect them, and sometimes they're coming and they are in a nice pile together. Does it go against this idea of entropy, or it was still work that put these leaves in a pile? When it was wind was blowing, and now we have these leaves, they were all over the place, now they're in one pile. Wind is motion of air. It's kinetic energy of moving air, wind. So definitely it's going to be some work. So it's not by itself. So we are going to talk about that more. So now your quiz about here. There